Good afternoon. Our rivers need to be assessed because there are several disturbances and alterations caused by humans, such as loss of riparian vegetation or presence of invasive species or channelization or pollution or transversal or longitudinal barriers that cause alterations in the aquatic communities as well as in the ecosystem functioning. So the Water Framework Directive says that we should measure the composition and abundance of biological communities, such as invertebrates, diatoms, other plants, or fish. Traditionally, we collect the samples and then we identify all the taxa under a microscope or stereo microscope, which is quite complex and time consuming and requires large human resources, costs, and high taxonomic expertise. In alternative, DNA metabar coding combined with high throughput sequencing from bulk samples or environmental samples provide information that can be converted into operational taxonomic units or can be used as individual sequence units or exact sequence variants. This is a quicker method and maybe less expensive, but definitely it avoids the sorting step and the counting of individuals. In both cases, we'll have a list. In the first case, a list of taxa, and then we can apply an index, well-established indices, to calculate a score, and then the ecological quality ratio by dividing that score by the reference value for the river type. Finally, we calculate ecological status by looking at a classification system. When we have the molecular information, for example, lists of O2s, we can also convert this list into a list of taxa. However, this list of taxa is not the same as the one above, even if they are both from the same sites and from the same sample. And that's because databases are not complete and we have differences in the detection of species uh, by both methods and also differences in detection of abundance. Therefore, by using this new list of taxa, taxa B, we will calculate a different score too, so score B. This score B, to obtain an ecological quality ratio, we also divide it by the reference value for the adequate river type. However, this reference value is the same. So we will introduce here an error or another error, another difference that may lead to a different classification of the same river site. So to apply this method, we would need a molecular based reference value, which is something that usually we don't have. Um, Looking at what was done before, so we divided our rivers into types, and these types were built based on information such as temperature, air temperature, precipitation, altitude, geology, drainage area, or size of the river. And after that, for each of these river types, we used the reference, the values from the samples collected in these river types and analyzed their EQRs and calculated a median value, for example, of these EQRs for a given type. And that's what we use nowadays as a reference value for a metric or an index. So if we want to use the molecular information, we should rebuild this entire system and calculate these reference values for based on the molecular data, because as we saw, they are different from when we perform just the morphological identification. However, we can consider other possibilities and think that a river is a continuum and not really divided in pieces as we consider in the typology. So, for example, this area that is between two types of rivers, this type red and this type blue, is in fact an intermediate, has in fact intermediate characteristics. So we'll have there also probably also populations that have intermediate characteristics. So we may have a new taxa, but the sum of the other ones from the red side should be there also. And we may have less individuals of certain taxa or the same from other. So what we could do is to calculate site-specific reference conditions in all the cases. Whenever we want to assess a site, we will calculate the reference condition adequate to that site and not really to the river type. Um, so that's what we try to do here with this approach, the Hydra. That is a predictive modeling tool that allows the prediction of biological entities based on environmental information of sites. I say biological entities because we already tested the approach with taxa, for example, invertebrate taxa, and we also tested this approach more recently with OTUs and diatom OTUs. So the Hydra is a combination, in fact, of three machine learning techniques: support vector machines, multi-layer perceptron, and k-nearest neighbor. All of them work in different ways. So the multi-layer perceptron is more like a neural network, but they all allow the prediction of biological entities by the similarity in the support vector machines, the similarity of a site, the environmental similarity with other reference sites, in this case with a group of reference sites, in this case with the reference sites that are more close in terms of environmental characteristics to the our test site, independently of in which river type or which group it would be. In the multi-layer perceptron, um, we train the model by giving a lot of information, input information, biological and environmental, and we say, okay, with this biological and environmental data, our output is this output. And so afterwards, we are able to test a different site, and it will give us the adequate predictions in terms of biological entities if the model works well. So that's what we did. So we trained um, an Hydra model with reference sites from our study area, and we give also the environmental training data. These environmental training data must be of only non-pressure variables. So it's typological information and not variables such as the nutrients or the land use and so on that translate disturbance. And then we will obtain the biological entities predicted in that site, and by summing the probabilities of biological entities occurrence, we will obtain an expected taxa under reference condition. And by using that expected taxa, we can calculate the ecological quality ratio or the observed expected taxa um, for that sample. And finally, we can calculate a reference CQR, so the median of all the reference sites. Based on the reference site CQRs, we can also build a new classification scale, usually between 0 and 1, or theoretically between 0 and 1. And the I good boundary is usually calculated based on the value for the 25th percentile of reference sites. Uh, in step 2, the model validation, we will use some reference sites that weren't used previously to build the model to test the model. So, following the same scheme, we should obtain a classification for these validation sites of high or good, and not less, because they are reference sites. This is a way of validating the tool. Finally, for the test sites so sites of unknown quality, we provide the biological data from the samples, we provide environmental characteristics, and we predict what should be there if the site was in reference condition, and then we compare with the observed. So we calculate the observed expected ratio or the ecological quality ratio and get a classification. We did that with 81 sites from central Portugal, where we followed both approaches. So we did a morphological identification of diatoms, and also we used the metabar coding to obtain the lists of OTUs from all sites. From these 81 sites, 44 were reference sites, and 37 disturbed according to abiotic data. So habitat quality, oxygenation, land use, connectivity, riparian vegetation, all these was used to separate the 44 reference sites. And we obtained a total of more than 1,000 OTUs and more than 300 diatom species in the 81 sites. And in the reference sites, we had 692 OTUs and 224 species. As you can see, we always have um, a third of species compared to OTUs. So we'll have really different lists from the start. Then in our model, we used variables such as predictive variables, um, such as elevation, mineralization, geology, lithology, channel width, besides the climatic variables. We can choose here whatever is more adequate to our biological element. It might be a different list, but we should use the typological variables. This to approximate our method to what is required in the Water Framework Directive. In this case, we decided to build two models, one based on species, the hydromorph, and the other based on OTUs, the hygiene. We excluded rare taxa in both cases, and all taxa in the models were predicted with an accuracy above 50%. And we built two new classification systems, one for species and another for OTUs. These models were quite accurate, both of them. We also got good values in the cross-validation, and we were able to define the boundaries for all classes. Then we assessed our test sites. And again, we are looking into different things. When we convert the OTUs into species, we could see that looking just to the most representative entities in all classes, we could see that 50% of the genera were different and 30% of the species were different. So when we convert OTUs to species, we are not looking into the same lists. In terms of response to disturbance, both models were quite sensitive. They showed a better degradation gradient than the EPS even. When we look into the correlations with individual disturbances, we can see that hygiene responded, so had significant correlations with most of disturbance variables. and more than the idmorph, so the model based on morphology, and well, quite similar to the EPS. Um, all them responded, so were significantly correlated to a global disturbance gradient given by the PCA axis 1. However, the Igene had more significant responses than the idmorph. So, well, yes, we have differences. So when we calculate an index based on morphology, when we calculate index based on OTUs converted then into species, or when we when we build a model based on morphology or based on OTUs, we are always looking into different things. So we should know what we are using and measuring. That's the first idea, take home message. The models have the advantage of avoiding setting new reference values for river types, which is something that would need to be done for all countries. They also avoid the artificiality of type-based reference conditions. The models with the molecular data avoid also the problem of unassigned reads. We don't care about this conversion. We could just look into the molecular information. Also, so this may be a faster approach than the morphological one, a cheaper approach, and also maybe more sensitive. On the other hand, the machine learning models, they take time to be built and they require big data sets. Our data set was not that big, but we were looking into a small area. So if we were looking into the whole country, we would need really a big data set to make it, to have good accuracies in the prediction of OTUs or taxa. Um, we don't use here abundance data. So this is something that is not aligned with the Water Framework Directive, because we think that these techniques, especially the multi-layer perceptron, do not work very well with the abundance data. However, there wasn't a difference in using abundance data with EPS or not using abundance data with IgG, for example. So we didn't see a disadvantage in not using the abundance data. We could also try other techniques, other modeling techniques, um, as probably there are others that may be more efficient with abundance data, more to be more compliant with Water Framework Directive. Finally, it's still difficult to give an ecological meaning to our results in terms of OTUs, because we don't know the meaning of an OTU in terms of response to this or that environmental characteristic, which is something that we usually discuss along with the classification. But this is something that could be investigated. Um, so this I said already, and we could also test the use of alternative molecular data, such as the individual sequence units or the exact sequence variants, in order to um, have more meaningful ecological um, outputs. This is something that we can test with these models. So I hope you liked it. Thanks for your attention. I'd like to thank the organizers of the meeting for inviting me. Also the organizers of WP2, Maria and Jan, and to thank my partners in these works, Salme Almeida and Andrea from the University of Aveiro, Sonia in Coimbra, and Agnes, Fred, and Valentin from INRA Tournon.